Once you have produced a protein model using X-ray crystallography, the data is published in a protein bank. However, these models may not always be entirely accurate, so we need to consider how well our model actually describes what the protein might look like in its native environment. Today, we're going to look at 5 features that you must be aware of when using models from the data bank. The first question we need to take into consideration is how much detail can we see? The resolution of our data is how much detail we can see. This is defined by the smallest distance between two objects at which they can be distinguished as individual objects. If the model has low resolution, it's not possible to accurately locate individual atoms. Therefore, it may not be an accurate description of what the protein is like in its native environment. With increasing resolution, it does become more accurate. The maps become clearer, showing separated peaks corresponding to the positions of individual atoms. Here you can see the increase in individual atom visibility from 1 angstrom to 3 angstroms. The second question we need to ask is whether we can see all the hydrogen atoms present in our protein. Hydrogen atoms are very small, so have a weak X-ray diffraction. Therefore, they can only be seen and localized if a high resolution is used. Therefore, in most protein databank files, the hydrogen atoms are absent. However, we know there is hydrogen all throughout the protein. Using the known bond length and angles of the heavier atoms it's bound to, the precise location of the hydrogen atoms can be determined. Hydrogen can be modelled in and more accurately described what the protein is like in its natural environment. Another thing to consider is that proteins are not static molecules. They are constantly changing conformation due to not only thermal and kinetic energy, but also binding of ligands and other molecules. This will be discussed later. A B factor is used to give a numerical value for the flexibility of certain atoms about their average positions within a protein. You can even colour your protein map according to atom B factor to reveal which parts of your protein are more flexible than others, like this galactose-glucose binding protein. How cool! This also allows you to identify side chains in an active site. You must be aware of the flexible parts of your protein when comparing it to your model, as there will be multiple conformations. There may be atoms in your model that are in a different position in the protein or atoms that are in 2 different positions. Don't worry, this is just due to its flexibility. Creating a B-factor map will help you to visualize this better. When determining the structure of the protein using X-ray crystallography, we determine the asymmetrical unit. However, this unit can have more than one copy of the protein present, and these may have different conformations depending on where they were in the catalytic cycle when the crystal was formed. For instance, a crystal may form with 4 different variations of the protein in the asymmetrical unit. One may be bound to the substrate, one to the cleaved substrate, one that isn't bound at all, and another to the final product of the enzyme's reaction. Technically, all of these variations are the correct structure of the protein, and this occurrence illustrates the dynamic nature of proteins. Once you've worked out the protein structure, the electron density that is unassigned relates to individual atoms or molecules, such as metal ions and water. These affect the shape and function of the protein. As with hydrogen atoms, a high resolution is needed to see small metal ions, such as sodium, magnesium or water. However, it can still be difficult to distinguish between them. It is important to consider the presence of water molecules in our model, as they can aid ligand binding. This is due to their ability to form hydrogen bonds between the ligand and the amino acid residues at the site of attachment on the protein. This facilitates tight binding and so affects the activity of the protein. Stable ligands are more easily detected through X-ray crystallography, as you can simply soak the crystal in a solution of the ligand, or just grow the crystal with the ligand bound to the protein. Unstable ligands are more difficult to detect, as you need to know complicated things that we won't even go into, because to be honest, we aren't even sure what they are. For all of you keen beans out there, check out this article that explains all this in further detail. Warning, it contains a lot of complicated words. I hope you've enjoyed learning about what features you need to look out for when taking models from the database. Please follow all our e-resource links to find out more.